So thank you all for attending and joining this 6th series of Eurotech Chemistry and Chemical Engineering speaker series. Today we have we are happy to introduce Professor Jérôme Wasser from Epfl, Switzerland, to join. He will begin the speech shortly. First, I would like to introduce Eurotech for those who don't know. We're an organization of 6 technical universities based in Europe and Israel. Technion from Israel, Epfl from Switzerland, TU Munich from Germany, TU Eindhoven from the Netherlands, École Polytechnique, or EP Paris as it's called now, from France, and then the Technical University of Denmark. We are happy to introduce this 6th series of chemistry and chemical engineering, and I would like to introduce Jérôme Wasser, who will now take the floor. Thank you.
So thank you for the introduction and also for this invitation to speak at this Eurotech symposium. So I'm very pleased to be here today. And I guess my talk will be probably the first, I think, on the series in purely synthetic chemistry. So it will be probably something different from the previous talk. And I will speak today about our recent work in the last 5-6 years on merging hypervalent iodine chemistry and Photoreduct chemistry. So in the first part, I will give a first introduction on how we came to this field and why it's interesting. And then I will focus basically on 3 stories: how we could convert carboxylic acids towards alkynes, and then how we extended this type of chemistry beyond alkylation to develop other transformation process. And I finished with a recent project where we moved to the defunctionalization of alkenes. But first, a little bit an overview of what is going on in my group. So basically, my group in synthetic chemistry is trying to develop new disconnection, making new ways to connect bonds together. And one big part of our research has been to kind of changing what the textbook tells us. So making what is called an Umpolung in the honor of Dieter Seebach from ETH Zurich that pioneerized this concept of changing, in fact, the reactivity of functional groups to introducing in the reverse way that is natural. And I will speak today of several of these reagents, in particular the alkynyl reagent. But my group is also working in other fields. In particular, we are working on the small ring activation, strain ring, a nitrogen substituted strain ring to make bioactive compound, and also smart tether in catalysis to functionalize alkenes. So unfortunately, I will not have time for this topic today, so I will need to give that as another occasion. So let's start now with a short introduction on how we came to the discovery of this ABX reagent. And our chemistry was based on the question of how do you make alkynes. So alkynes nowadays are very important in synthetic chemistry, also in chemical biology and in materials. And if you look on how you transfer an alkyne group, it's nearly always based on the negatively charged acetylide. If you would like to transfer your alkyne to a nucleophile, and there is a lot of nucleophiles, especially if you think about natural product, bioactive compound, proteins, you will need to have an electrophilic alkyne syntones. And what you do normally in synthetic chemistry, you just add a substituent like an iodine to make it a good leaving group. But in the case of the alkyne, because of the sp hybridization at the C carbon, that is not enough, and you need to have a better leaving group. And that's why you get interested in this hypervalent iodine reagent. They are, for fundamental chemistry, very interesting because you are breaking the octet rule around the iodine. We have now up to 10 or 12 electrons around the iodine. This is due to this hypervalent bond that you can describe as an overlap of the p orbitals. You have one binding orbital, one non-binding orbitals, and in fact, 2 of the 4 electrons involved in this bond are non-binding. So you can roughly say that the bond order is only 0.5, and these bonds are very reactive, and they allow you to do new chemistry to make these connections that are else not possible. So this field has been started in the 60s by Behringer, and has known a kind of golden time in the 1890s with researchers like Stang, Oshia, and Duncan. And then he kind of calmed down. And when we started in 2007, we wanted to use this reagent to make new transformation in synthetic chemistry. And the first reaction you worked on was this alkynylation of indoles. The indoles is one of the most important heterocycle. It's very important in medicinal chemistry, and coupling it directly with an alkyne was not possible at that time. And we reasoned that it would be possible just by using palladium or copper catalysis that were pioneered by Matthew Gant and Melanie Sanford. We could just connect these two building blocks together and access the alkynyl indoles. We tried a lot at the beginning, and these never worked. One key point that you have with this reagent is that they are unstable. So we needed to find other type of reagent that would be more stable. And that's where we started our research in this cyclic hypervalent iodine reagent. By incorporating the iodine into the cyclic structure, you enhance the stability, you stabilize the iodine by transfer of electron for the pi system, and these reagents are now much more stable. These reagents had been synthesized in the 90s by Ocean's Duncan, but had never been used in synthetic chemistry. In contrast to the CF3 reagent introduced by my colleague Antonio Toni at DH Zurich, that is now one of the most successful trifluoromethylation reactions in synthetic chemistry. And we found that indeed, these reagents allow you now to make this synthetic transformation using a little bit of gold catalyst at room temperature under air. It's highly selective reactions. So first question, before I speak about application, is how easy it is to get this reagent. So the original Duncan protocol was working pretty well. So it's an oxidation with periodate to get to iodine 3, and then an alkyne transfer. So it's only 2 steps, 3 if you count the preparation of the cellulated alkyne that is not commercially available. And now this reagent is commercially available by several companies. But recently, we also improved this protocol. So we have now 1-step procedure using only commercially available reagents. So you can really make in two days of work, 40-gram batch of these reagents. So they are really easily accessible, and they are also relatively stable. So we then, over the year, used this reagent in many different transformations. So we started with CH functionalization, then extended to alkene functionalization, simple nucleophile functionalization, carbene functionalization. And for the talk of today, I decided to discuss the functionalization of radical, especially using Photoreduct chemistry, as is a very intensively investigated field of synthetic chemistry nowadays, because there is mild method now to generate this radical using Photoreduct chemistry. There is naturally other researchers that were interested in this type of reagent. Okay, seems. Sorry, my screen seems to have frozen. That's naturally always the net things. Yeah. Okay. And these researchers further developed the method with this reagent, and we recently reviewed the field in 2016, but there is now many transformations using this type of reagent. Now, what is the connection to Photoreduct chemistry? So there is really a problem of connection, because I will maybe restart the presentation because it seems to always freeze, or maybe it's only a problem of that. Like that. I think the laser pointer was slowing down. So with the normal pointer, it should be still visible. Yes. So the fundamental of Photoreduct chemistry is to activate organic compound with light. And if you activate organic compound with light, you move after intersystem crossing to the activated triplet system. And what is very simple in there is that in the same time, you make a stronger reductant and a stronger oxidant. And these are surprisingly used only very late in synthetic organic chemistry. Naturally, our colleagues working on energy conversion was using that much earlier to absorb the light and transform them into electricity. But in synthetic organic chemistry, there was already work in the 80s, but it's around 2008 that the researchers really started to get intensive. When people realize you can use this type of chemistry in 2 types of catalytic cycle, You can have a reductive catalytic cycle where a donor is oxidized, or oxidative catalytic cycle where an acceptor is reduced. And naturally, you need then to close the catalytic cycle in the ground state by going back to the starting oxidation state of the catalyst. And we are naturally thinking that our hypervalent iodine reagent being good oxidant, they should be perfect to interact in this as an acceptor or to intercept this radical intermediate formed during this transformation. And there was a lot of possibility at the interface of Photoreduct of hypervalent iodine reagent, considering in fact the rich redox chemistry of hypervalent iodine. And the first project I wanted to speak about is the transformation of carboxylic acid to alkynes. Conceptually, it's a very interesting transformation because the carboxylic acid are coming from the biomass that are broadly available, and the alkynes are one of the most useful synthetic compounds for making drugs and materials. So there is a big interest to convert carboxylic acid to alkynes. So the students that work on this project were Frank and Marion that started the Photoreduct chemistry, and now the project is continued with Stephanie and Diana. The breakthrough came, in fact, first from another group, the Li group in China, that realized that these alkynyl hypervalent iodine reagents are excellent quenching reagent for radicals. They generate the radical from a carboxylic acid using a classical condition, strong oxidant and silver, to get the alkynes. So this works very efficiently, but there are still several limitations if you consider also the concept of green chemistry. You need quite a lot of silver catalyst, and you have this very strong persulfate under heating condition. So that was a little not satisfying condition for this transformation. There was a first example of Photoreduct chemistry published by Yun Chen that used ruthenium catalyst blue lead and alkynyl sulfone to trap the radicals, but he was using a reductive method where you need to first activate the carboxylic acid to make it under an ester that can be then reduced. So it was a little bit less attractive because it was a two-step procedure. So we started there with the goal of using a direct method using Photoreduct chemistry to transform the acid into the alkyne. So if you think about the design of the catalytic cycle, what we need to realize there is first activate the metal with light to get a stronger oxidant. At this stage, we would be able to oxidize the carboxylic acid to the radical that will lose CO2 to generate the carbon-centered radical. And the carbon-centered radical will then react with the alkyne to generate the addition product. The addition product will then eliminate the iodosyl radical that you can draw into resonance structure to generate the alkylated product and this iodosyl radical. So up to that, everything was known in the literature because especially the Macmillan group has studied a lot this decarboxylation with Photoreduct. And the Li result indicated that the alkynyl reagent can react with these carbon-centered radical. But the key question was now, would we be able to close the catalytic cycle and then transfer the electron back to the radical, reduce the radical to the carboxylate, and get back the ground state of the catalyst to make turnover? And there is some indication that you can use based on thermodynamic. You know that for oxidizing the carboxylate, you need a catalyst that has at least this reduction potential. And that can guide you in the choice of catalyst. The other value here was naturally not known, so we could calculate it. And we found out that this type of radical is a relatively strong oxidant that should be able, in fact, to allow turnover in many of these catalysts. So here is the initial catalyst screening we did, and you can look at the redox potential, so the oxidation state in the activated state, and then the reduction on the ground state. And you quickly realize that many of these catalysts are not strong enough oxidants to promote the transformation. And in fact, if you use the catalyst, you will just re-isolate your starting carboxylic acid. In contrast, all the other catalysts are strong enough reductant to make turnover, but in reality, only one of these catalysts worked. And this is this iridium catalyst that has a very good balance between oxidation and reducing power. And it's probably why it's so successful. You see many, many applications of this type of iridium catalyst in synthetic chemistry. So these one work then perfectly and give us now 90% yield of the desired product. I will not today focus a lot on the scope because I think it's more interesting in a presentation, focus a little bit more on the principles. But this reaction works very well for different carboxylic acid. being amino acid, oxy acid, and it also works for aliphatic carboxylic acid, although the yields are slightly lower in this case. And you can make silyl alkyne and also aryl alkyne. And I have to say it's a very competitive field, so we were back to back with the group of Xiao that published a very similar methodology at the same time that we were out of this methodology. So after this proof of concept, we wanted to go into several directions. And the first question we ask us is, can we use other way to generate the radical instead of decarboxylation? And one inspiration we get is the radical fragmentation based on the fundamental work of Zamir Zard. He developed this reaction where you cleave this NS bond under classical radical condition. You then get a fragmentation, ring opening due to ring strain. And you get then this alkyl radical that can be intercepted in a Gizeh reaction. And in the time we were working on this project, two other groups, Xiao and Leonori, developed Photoreduct-based approach for this fragmentation. You could either use reduction, and then you will make an addition to an alkyne, but in this case, you get the alkene at the end of the transformation. Or you could make an oxidation of this carboxylic acid that will then fragment, losing CO2 and acetone to generate the same radical. And then you get to the functionalization of the alkyl radical. This method was reported by Leonori, but he was able only to make CX bond formation with heteroatoms and no CC bond formation. So we wondered if our reagent would be now able to introduce this carbon alkyne bond. Maybe a technical note, to synthesize these oximes, we needed to have this hydroxylamine that is very expensive. And the only way to synthesize it was published in a Chinese patent in Chinese only. So we first optimized the synthesis and developed a reliable protocol. So now we can get really big quantities of this oxime in good yield. And the condensation is working very well to get this type of static material. So it's a very reliable protocol to prepare these activated oximes. The preliminary result using Leonori condition were promising. So we get about 50% conversion and 20% of yield for this transformation. And we decide again to focus on the catalyst design to see if we could find a better catalyst for this type of transformation. So if you think a little bit again on your reduction potential, so the proline that we use in the previous project is here. And here is the iridium catalyst that we was very successful. And here is the Fukuzomi dye used by Leonori. And if you look on the oxime substrate, is about in the middle here at 0.15 volt. So we were thinking that there should be a more optimal catalyst that is not as strong as the Fukuzomi dye, but it should be stronger than the iridium catalyst. And that's where we become interesting in the carbazole type of catalyst. So in this year, there was only 1 or 2 publications by Zheng using this type of catalyst. And they have also a very nice balance of oxidating power and reductive power, and are among the most stable organocatalysts. Still not as good as the iridium, but still much more stable than other organocatalysts and with a good lifetime of the excited states. And these catalysts, in fact, give us promising results, still not complete conversion, but a very clean conversion, a very clean reaction with already 50% of yield. So we wondered how we could make this catalyst a stronger oxidant. And again, the design of the catalyst is pretty easy. You introduce an electron withdrawing group on the carbazole, and you have then changed the redox property. It becomes a stronger oxidant, slightly weaker reductant, but still reducing enough. And in fact, now we can get full conversion on the good yield in the desired transformation. So this catalyst was synthesized, in fact, by the first time by the group of Bunz, more for OLED applications, so not really Photoredox, and is easy to synthesize in one step from commercially available compound. So with the common group, we could calculate the ESP potential of this compound. They are pretty close from CZ-IPN on the chloro derivative. What is interesting in the chloro derivative that is not anymore completely symmetrical, so we have some halogen-halogen interaction that break the symmetry. And this was the predicted structures, and we were happy to see that the X-ray of this compound confirmed, in fact, that you have lost this symmetry and that there is this interaction between the halogen and the carbazole rings. So within my group, the students take the habit to give nickname to these different reagents. And this one was called the dragon. maybe if you have a lot of imagination, you can see the dragon in the structure of the catalyst. But this catalyst in any way works very well to make this fragmentation reaction. And the interest in this transformation is that you get alkynyl nitrile that are a very good building block to make other heterocycles. So we could make a broad range of this derivative using now the chloro-CZ-IPN under this condition. The next step we wanted to do with this chemistry was to increase the complexity. So we have been very interested in my group to move towards more complex target, and especially peptides. So peptide drugs are on the rise. There is more and more need to be able to modify natural peptide to get new building block. And we were naturally interested to see if we could make a decarboxylation on the C-terminus of peptide, knowing that we have all the nice follow-up chemistry of alkyne, like cross-coupling, Whiskey cycloaddition. There is really a lot of potential for this reaction. The main issue we had when we started is that a small change in the substrate already make the reaction less efficient, and we get only very low yield in this transformation. And Marion was able to develop new conditions for this transformation using the organocatalyst, the CZ-IPN. And I think now in our group, the CZ-IPN has become the starting point for many new projects. It's really a good point to start. And using especially the aryl EBX. And from these 2 points, it's clear that it's the aryl EBX that is the key point. We know now that probably due to the stabilizing effect of the aromatic group on the radical intermediate, these reagents are much better radical trap than the silyl or the alkyl reagent. And they are now efficient enough to functionalize this type of radical. Using this transformation, we can transfer different aryl reagents, and the reaction is tolerating many functional groups like bromine, alkyne, Aziad. So we can really have a lot of functionality introduced into the dipeptide. We can make also other dipeptides than just the proline one. So this decarboxylation on proline is particularly easy. So you need always to see if the reaction is general and if you can go to other compounds. And we are pleased to see that other peptides derived from alanine, phenylalanine, or glycine are also able to react under this condition. We then increase even more the complexity and we move to tetrapeptide. So the tetrapeptides also give very good results, and we can tolerate again a lot of the amino acid that you have in proteins. We can tolerate asparagine. We can have also lysine. So the lysine is possible because the nitrogen is usually protonated. Histidine, and difficulties started with the easier to oxidize amino acid. For example, with tyrosine, you need to change the catalyst to a slightly less oxidizing catalyst to enable the functionalization of these substrates. Whereas tryptophan really didn't work under this reaction condition. You can protect it if you want to make it to work. Then if you have an electron withdrawing group on the tryptophan, then it's working again. But if you have an unprotected tryptophan, it's oxidized faster than the C-terminus, and then the reaction will not work anymore. What is very interesting with this reaction is that you can distinguish between terminal and side carboxylic acid. So the aspartic acid on the side chain is not touched, and you only decarboxylate at the proline position, the C-terminus position, to get the alkyne at the C-terminus position. I didn't present it today, but the alkynylation reagent reacts spontaneously in absence of any catalyst with thiol. So if you have a free cysteine, what happened now is that we will first alkynylate the sulfur, and then we'll alkynylate the C-terminus. So we made kind of provisory protection of the thiol. So naturally, an important question was if the reaction was limited to proline, or if other amino acids of the C-terminus would also be tolerated. And we were happy to see that you can tolerate a lot of other amino acids: terminal phenylalanine, glycine, serine, histidine, methionine. There is a small limitation, like tyrosine and aspargic acid doesn't work as well as the other amino acid, and tryptophan doesn't work at all. So for tetramer, this reaction is working very well, and we can make a lot of derivatives. The weakness of this chemistry is now when you start to increase the size and you move to hexamers or decamers, the yields start to drop. And if you go to protein, you don't see any reaction anymore. So this is naturally a big drawback at the moment. And we're thinking on way to improve this reaction on larger peptide. What we already try is to put the catalyst on the peptide to see if you can get a proximity effect, but this didn't really improve the transformation. So we are still working on this issue to really functionalize larger biomolecules. So that's what we are on this carboxylic acid derivative. We are also now working on methods to transfer alcohol to alkynes, but is not yet completely ready to present. So what I would like now to move on is the question is, can we do other transformation that alkyne transfer using this principle of combining Photoreduct chemistry and hypervalent iodine reagent? On this project I will now present, we also start with Frank and Marion, and then two postdocs, Sébastien Alazé and Johannes Preindl, contributed to this project, and now it's continuing by two PhD students, Raphael and Elliot. There is naturally other hypervalent iodine reagents that have similar structure that have been reported in the past. Most of them have been done by Victor Duncan, that is really a fundamental inorganic chemist working on the development of new hypervalent iodine reagent. And two were particularly attractive for us, namely the Aziad and the cyanide-based reagent, because Aziad and cyanide are among the most useful functional group in synthetic chemistry. And there is only a limited range of reagents for transferring these groups. All of them are highly toxic and have other safety issues like explosive behavior. So when you go from a carbon-based reagent to a nitrogen reagent, a comment we get sometimes where you submit a publication is that you are doing always the same. But this is a wrong assumption. In fact, a carbon is not a nitrogen, and there is many properties that are different. I just want to point on two. One is the ion electronegative of nitrogen, and this will have a very strong impact on the oxidation potential of the reagent, as we will see. The second is the bond strength. Just as an approximation, I show the carbon-hydrogen bond strengths, and you see that the carbon-hydrogen bond on alkyne and on cyanide are extremely strong. The hydrogen-nitrogen bond on Aziad, in contrast, is very weak. So that means it's much easier to generate the azido radical than it is to generate a cyano or an alkenyl radical. That will also completely change the type of chemistry you can do using this reagent. The first question we ask us is, can we move from alkynylation that I just presented before to cyanation? And here what happens when you use the same condition for this transformation, the reaction works somewhat, but there is a new side product that we never observed before, probably resulting from the addition of water, as we will see later in the mechanistic discussion. And we begin to see cross anhydride emerge from the functionalization of the carboxylic acid. Fortunately, we could stop this type of side reaction by diminishing the amount of base and by using molecular sieves to remove all water from the media. And then we can get good yield of this addition product. We move also to THF as a solvent. We see a little bit of cyanation of the solvent, but still THF is giving the best result from all solvent. And to show you a little bit the superiority of the hypervalent iodine, if you use a classic reagent based on sulfur, you get only very low yield of this desired product. So this reaction is especially powerful for the cyanation of amino acid. So it's working very well for proline derivative, but also non-cyclic amino acid, glycine, alanine. You can even make more challenging one like methionine or lysine, and it's also working for dipeptide. Again, not for tryptophan. You can also make oxoacid, but what you cannot do is aliphatic acid. And this was a little bit surprising if you consider that the two reactions should have the same mechanism. Why was one working for the aliphatic acid and the second one was not working? So at this point, we decided to study a little bit more in detail the reaction mechanism. And we first tried to establish if you really have a radical in this transformation. So for that, we turn to the radical clocks. So you can observe in both cases a radical opening. So this is a well-established cyclopropane radical clock, and it opened to form the product. And this one was designed to be more similar to our substrate. And again, the ring opening was observed. So the aldehyde was probably observed after hydrolysis of the enamine that would be formed after ring opening. For the cyanide, we didn't observe the radical trap didn't work, but we observed with tempo the adduct that was formed after decarboxylation. Another important question that I see that has been asked to me many times is with this very reactive hypervalent iodine, it's surprising that the activated photocatalyst doesn't react with the reagent. On that, we can do using these Stern-Folmer studies. In the Stern-Folmer studies, we will observe the fluorescence of the catalyst in presence of different substrate and reagent you have in the reaction mixture, and you will see if there is an effect, and that will tell you if the catalyst is reacting with one of these reagents. And here you can see very well that you have a linear relationship of the fluorescence with the amount of carboxylated added, but you have no effect of added reagent. So that really tell you that the reagent is oxidizing the carboxylate and is not reducing the alkynyl reagent. The second question is that we could also imagine to have a radical chain process where the photocatalyst is only used as initiator, and then you don't have any more catalytic cycle. So we measured the quantum yield of this reaction, and I don't think it's a very strong experiment. The quantum yield was below 100%. Unfortunately, it could be a bad radical chain. So if it's over 100%, you are sure you have a radical chain. Below 100%, it just tells you that both catalytic cycle and radical chain are possible. So to get better insight into what was happening for these two transformations, we turned then to computation that were performed by Matt Vodrich at our institute. And we first computed the interaction with the radical, with the hypervalent iodine reagent. The radical can make, as a first possibility, an alpha addition. And it's interesting to see that the iodine bond is so weak that you never see an intermediate. You have a concerted addition dissociation to get directly to the product. But there is also a second possibility with the radical. The radical could make a beta addition, so on the second atom of the iodine. On this stage, you generate this time an intermediate that we could locate via computational chemistry. But again, this intermediate is not very stable. You make a very fast 1,2 silicium shift to get to the product on the iodobenzyl radical. Then we were wondering about another reaction pathway where the oxidant will oxidize the radical to the iminium and then make simply a nucleophilic addition. So the oxidation of the radical by the reagent is possible. It's endothermic, but not too much. So it's possible that you get a little bit of oxidation taking place. However, this radical anion is pretty stable, and it's very difficult to collapse into the anion on the iminium that then recombine to give the observed product. So these indicate that the most probable pathway for the alkynylation is really a radical pathway. Naturally, what is now interesting is to compare what happened when you replace the alkyne with a cyanide. So we did first the computation of the alpha addition pathway with the cyanide. So this is working fine, and is in fact a reasonable pathway for the alkynylation of the cyanation of the radical. And if you look on beta addition, the start looks fine, but then it's higher in energy. And especially the major difference, you will then get an isonitrile instead of a nitrile. On this one, we'll have big difficulties to rearrange to the nitrile product. So we see the energy barrier is now close to 33 kilocalories, and we never observed an isonitrile. And now comes the interesting pathway, the single electron transfer. It is now easy to see that the cyano reagent is a much stronger oxidant. So it can now oxidize the radical, and it's not only slightly endothermic, only 2 kilocalories to get the iminium. And now this radical anion is highly unstable and dissociate easily into the cyanide and the radical, iodosyl radical, and both can then recombine without barrier to give to the product. And we see now that changing the alkynyl to a cyanide, we have completely stitched the reaction pathway, and we are going now to a double set transfer to generate the iminium. And that explains very well the difference between the two transformations. So this iminium can be intercepted by water, and with aliphatic carboxylic acid, the second set is probably not occurring, what explains why the reaction doesn't work so well. And recently, we wanted to exploit a little bit this two-set pathway to develop other transformation beyond cyanation. And I have to say, we didn't invent naturally this type of oxidation of carboxylic acid on peptide on amino acid, it's very well established that you can do that using the so-called uppermost process using electrochemistry. And there was beautiful work of Dieter Seebach in the 80s. In fact, we're doing the PhD of Philippe Renaud, where he did this oxidation using electrochemical oxidation to get either the NO-acetal based on acetic acid or methanol. And more recently, Lara Malins further developed this chemistry. But we were interested to know if we could do this reaction under photochemical condition instead of electrochemical condition. And we had the first success using simple acetoxy benzodoxolone as an oxidant and CZ-IPN as a catalyst. But this derivative is not isolable, it's very reactive. And what you isolate after column chromatography is the water derivative. So what we did is we just did the reaction in presence of methanol, and now we can get a high yield of the NO-acetal, and 76% yield can be isolated. The reaction is very efficient. We need only 2 equivalents of methanol. And in this case, the classical ruthenium catalyst was slightly better than the CZ-IPN, although the CZ-IPN also gives you the product. So you can add different types of alcohol, and that's maybe a big advantage compared to electrochemistry, where you often use a large excess of alcohols. We need only 2 equivalents, and we can add different types of primary alcohol and tolerate functional group like alkyne or Aziad. We can also add more complex alcohol, especially the one derived from serine or threonine, and we can perform the reaction on other amino acids than proline, like alanine or phenylalanine or valine. So this transformation is working pretty well. However, a drawback of this transformation is in the stability of the product. These NO-acetal are not so stable. They can slowly decompose. You need to be very careful when you purify them. So it will be more exciting to be able to do carbon-carbon bond formation to get more stable product. So the idea was now that we will make use of this very active intermediate to intercept it with other nucleophile. And the first reaction we developed was now a Friedel-Crafts transformation with phenol. So this transformation is activated by boron trifluoride as Lewis acid. And you can then add different type of phenol to this intermediate. Simple phenol, electron-rich phenol are working pretty well. Electron-poor phenols are too slow in this transformation. You can add also more complex phenols like the one derived from steroid. And you can use other amino acids like alanine or glycine. The interest with glycine is because you get a more reactive iminium intermediate. You can also add less reactive phenol like fluorinated phenols. And what is also interesting is that you can use the natural nucleophile that you have a peptide, like the tyrosine derivatives, and you can now make cross-coupling different peptides via bonds that are not found in nature. So it's like new non-natural peptide can be done using this methodology. So after we have done phenol, we are naturally interested to do the second natural nucleophile that you have in biology, indole. But the indole doesn't work with boron trifluoridate. So the best condition to make the reaction with indole was the use then of TFA, simply trifluoroacetic acid, to make this transformation. With this reaction with indole, we can again make different transfer of indole. We can even transfer it to tetrapeptide on different type of amino acid like alanine or valine. And what is interesting is if you have a C3 substituted indole, then the C2 position is not reactive anymore, and you get an N-acetal instead. And again, you can do this transformation even on more complex tryptophan-derived peptide to get new cross-coupling of peptide. So with that, I want to move on to the different type of reagent, namely the Aziad-based reagent. So the Aziad-based reagent is a completely different story. This reagent is now very sensitive to redox active system. It is especially very easy to reduce it to generate the carboxylate on the Aziad reagent. And this has been demonstrated by different groups, Armido Studer, Cristina Nevado, and Michael Greeny, for example, that you can use either metal catalyst or Photoreduct approach to reduce this reagent. So we use this transformation in azidolactonization that we developed in our group to generate the azido radical and then cyclize onto the generate intermediate using this time a copper photocatalyst that is a very strong reductant. But what I want to focus in this transformation is again showing how the reagent design is important in this case, because this reaction works very well with the carboxy-based reagent. But if you move to the dimethyl-based reagent, you see only traces of the product. The main product was this ketone, and we later find out that the ketone was coming from the alpha azido compound when you make the reaction under perfectly dried condition. We found later that the best catalyst was not copper, but palladium Lewis acid, although the reaction is also working with other Lewis acid like tin and zinc, and you can get different alpha azido compound in good yield using this transformation. So what is occurring in this transformation? So in the copper DAP-based chemistry, copper is a classical photoredox based to reduce the organic substrate. So we will generate the azido radical. The azido radical will add to the double bond, and you get now the tertiary radical. Because of the proximity of this aryl group, it's easy to oxidize this radical to the carbocation, and then you can cyclize to get the lactone. On the other hand, the dimethyl reagent is not a strong enough oxidant to be reduced by the copper catalyst. It will just be activated by the Lewis acid to generate a iodonium. The Eudonium chemistry is then a classical chemistry where you activate the double bond with the iodonium. You cyclize to make the lactone, and this intermediate is a very reactive because of the good leaving group activity of the iodine. You can make a 1-2 shift of the phenyl, and you quench the intermediate with the azide to get the observed alpha azido compound. So these show you that by small change in the reagent, you can get completely divergent reactivity. And we then developed an azidative ring extension where you generate the carbocation and get a 1-2 shift. But where we were in the process of making this transformation, we get in fact the only serious accident that we had in my lab. When we tried to make extra dry pure Dunkin reagent, the reagent spontaneously exploded and we get some serious end injuries. So we decided to study a little bit more in detail the safety profile of this reagent. And we found out, in fact, that this Dunkin reagent that is used a lot, in fact, around the world is quite a dangerous reagent because it's sensitive to friction and to impact. So we couldn't determine the impact sensitivity because it was too low on the crystalline reagent. So even small shock can make an explosion. Fortunately, already a third butyl group removed the friction sensitivity and make it much less impact sensitive. And finally, when you add a tosyl group on to replace the oxygen, you have a much more stable that is now pretty safe to use. And you can see that also from the DSC profile, you have a much higher thermal stability with the new azido reagent. And naturally, you need to see if you still have the reactivity. And this reagent is working beautifully to generate azido radical for the shift. And I think now this is a much safer reagent to use, but still, most people prefer to use the more known Dunkin reagent, although this one is an as good reagent to generate azido radical for the transferring azide to organic compound or biomolecule as we are studying now. And I will now finish with a short project on our first project on the functionalization of alkene using radical chemistry. So this project has been done by Stephanie and Stefano. And the idea is based also on a comment that we get a lot when we do this type of chemistry. They always tell that either you take a nucleophile or nucleophilic radical, what you always obtain is the iodobenzoate as a waste. You can naturally recycle easily, so it's not too bad. And in an ideal world, you will be able to make the reaction catalytic in this compound. So we are working on that, but it's still far to be efficient for the moment. But what we developed in the previous year is way to use this iodobenzoate to make multifunctionalization of carbene using copper catalyst and box ligand. So we can even make this reaction enantioselective. And we were able also to develop a 3-component transformation where you can use an external alcohol when switching from the benzodoxolone to the bis-3-fluoro derivative. So we were wondering if we could develop also multifunctionalization reaction based on radical chemistry. And here is our working plan. We wanted to work on enamide because we are always fascinated by this nitrogen-based building block. And enamide are naturally a rich history of double functionalization, where you can use an electrophile on the nucleophile onto the alkene to get these amine building blocks that are very useful for medicinal chemistry and synthetic chemistry. And recently, different groups have worked on the functionalization of enamine using radical chemistry, using an electrophilic radical, an oxidant on the nucleophile. You can make all these nice multifunctionalization reactions, especially the Masson group and the Guilezo group in France have published nice methodology in this respect. But we wanted to use another approach based on the work of David Nikiewicz. Nikiewicz demonstrated that you can make an unpooling of this type of enamide using oxidation, and then you could add a nucleophile on the radical trap to this intermediate to get a polarity inverse functionalization of the alkene. And naturally, with our reagent, we had the ideal tool in hand to do this type of transformation. So here is the preliminary result on this transformation. So we first try our starting point, the organocatalyst 4CZ-IPN. So the chemistry worked, so we have a good preliminary result, but the yield was still pretty low. So we moved to the more active dragon catalyst, and indeed we could increase the yield. And now came an issue that is probably pretty frequent in synthetic chemistry. We had a reproducibility issue, because the next time you perform this reaction with a new batch of reagent, we get a low yield on this transformation. So Stephanie take a lot of care to get extra pure reagent and develop a new synthetic and purification protocol. So now we have very high purity reagent. And we found out, in fact, that we still keep the same yield. So in fact, the low yield is the one of the pure reagent, and probably the whole batch was not pure. So once you know that the most probable impurity are iodine 3 precursors that are used to synthesize the reagent. So we screened different iodine 3 precursors, and the best result was obtained with acetoxybenzodoxolone again. So with 0.5 equivalent of this reagent, we now can reproduce the yield and get a good yield in this transformation. A slight optimization, and then we can get the reproducible yield in the synthetic procedure. And we can use this transformation for the functionalization of different enamides and carbamates. We can also use the reaction for the functionalization of enol ethers, and we can get multifunctionalized compound. And we can use different reagents to transfer the alkyne, and we'll see we have only aryl alkyne because we need, again, the highest efficiency possible that come with the arylated reagent. So to conclude, some comment on the mechanism of this transformation. So our speculative mechanism was based on the activation of the photocatalyst and then the oxidation of the enamine substrate. But do we have some experimental evidence for this step? We know what is the oxidation potential of the catalyst. What about the substrate? So the oxidation potential, the reduction potential of the substrate can be determined by cyclic voltammetry. And you see that for the mono-substituted N-carbamide and oxazolidinone, the reduction potential is about 1.2, 1.3. So it's really feasible to oxidize this substrate. Interestingly, if you put a methyl group at this position, it becomes much more difficult to oxidize this enamide. And in fact, this substrate in red doesn't work in our transformation. So it's really an indication that the first step could be oxidation of the enamide. There is naturally also the Sternfolmer experiment that is pretty speaking. So if you add the oxazolidinone to the catalyst solution, you see a quenching of the fluorescence, which indicates you could really get an oxidation as a first step. So once you are there, the next step would be an addition of the benzoate on the radical, and then a trapping of the radical with the hypervalent iodine reagent, and closing at the catalytic cycle, as we have seen in the previous project. Once you have closed the catalytic cycle, you generate the iodobenzoate that can add to the carbocation. So the question is, where is the acetoxybenzodoxolone important in this step? So it's not absolutely necessary. The reaction is working, but it's slow. So we think it kind of sustains the catalytic cycle by providing a larger amount of iodobenzyl radical so that the catalytic cycle can be kind of rescued if the reaction is too slow. It could also be acting directly as an oxidant in this transformation. Okay, so now we are also working on other multiple functionalization of olefins to increase, in fact, the diversity of product we can obtain. Okay, so I can come now to the conclusion of my talk. So what the picture I like when I think as synthetic chemistry. Synthetic chemistry is kind of a labyrinth today. So we want to make compound that we need for application, being in medicinal chemistry, in material chemistry, but still the syntheses are full of surprise and we often don't get the compound we want. So in my group, we try to develop new reactions that allow you in sort of a way to destroy the walls. You have this labyrinth to develop new transformation. So today I focus only on a single point, the interface of light with hypervalent iodine chemistry, but there are many other ways to make this compound, to activate this compound. And what is also interesting when you make reaction discovery, that sometimes you come out of your labyrinth in another point that was not your initial goal. And it's in fact conducted my group now to have several researchers working on the functionalization of biomolecules and proteins as a kind of non-expected outcome of reactivity discovery. So I'm part of Epfl. So the organic chemistry at ISIC is mostly 3 pure synthetic groups, Jiping Zhu, Nikolai Kramer, and myself. And we have also Xileu, an inorganic chemist that is also a very good synthetic chemist. We are in the island in Switzerland that is a little bit isolated from Europe politically, but geographically really in the center of Europe, and very nice campus next to the lake with a nice view on the Mont Blanc. And we also have a strong bioorganic department with Christian Heinis, Bert Fils, Imonai, and Pablo Rivera Fuentes. So I would like naturally to thank all my group that contribute to this research. So we needed to find a way to make a COVID-conform good picture. So we just take individual photo and make a patchwork. The advantage of that compared to the old style photos is that it's easy to update with the arrival of new group members because we can just add them once they arrived. So in red, you have again the name of the main contributor for this talk today. So they are really the one that did all the work in the lab. I need to thank of collaboration in theoretical chemistry, Professor Clémence Commonbeuf and Matt Woodrich, what I presented today. This funding agency for their support. I would like also to highlight especially the NCCR Catalysis, a new consortium in catalysis that just started in Switzerland with 40 research groups focusing on catalysis research. And I would like to thank again the organizer of the Eurotech Symposium for their invitation. And I will be very happy to answer any question you would have. Okay, thank you for your attention.
Thank you so much, Professor Jérôme Wasser. It was very, very interesting. And also for the diversity compared to the other presentations. I can see we already have one hand raised. Please feel free to ask Professor Wasser what you want, either in the chat or physically by turning on your camera and unmute you. Thank you.
Yeah, so I have one question regarding, I have a few questions regarding the decarboxylative radical generation that you showed. First of all, it was an excellent presentation and a nice piece of work. So I was wondering whether you can also try these reactions using neuraminic acid derivatives in case of sugars. So you have worked with peptide. So did you try with neuraminic acid derivatives as well?
I don't think we tried the normaric acid derivative. It's an oxo acid, if I remember correctly. So it should be possible to decarboxylate it. It should work pretty fine. Yes, we didn't try the substrate exactly. We are now more focusing on directly the alcohols because there's even more alcohols in sugar, but it is not yet completely developed. But yes, that could be an interesting substrate to work on. Yeah. But we didn't do it yet.
And after seeing your work on peptides, I was wondering if you can make substrates for Bergman cyclization, because probably that would be interesting to check if you can easily assemble 3 alkyne units and in the same pot, if you can get a Bergman cyclization going. So then it would be a good substrate for Bergman cyclization based antibiotics as well.
Yeah, so I was thinking how you would go to introduce, because for the Bergman cyclization, you need basically 2 alkynes on an alkenes, yeah? And I was wondering, so you would like to put these directly on the hypervalent iodine and transfer to the you need an indiene. So we didn't go so far as making indienes. In another project, we make inine. So we have an alkyne with an alkene on, but we didn't try this substrate in the radical chemistry, as long as a reminder. I think we tried this only in the carbene chemistry. And then making an indiene for Bergman cyclization, yes, could be interesting. Although I'm thinking there is probably other stuff that would be easier to introduce that would be phototoxic. Yeah. So that would be probably a higher level one, because the reagent would be probably a little bit unstable. I would need to think if it's possible to make it. Yeah.
And the last question is, in your mechanism where you showed your olefin was creating a radical cation, and that cation is getting trapped by your iodobenzoic acid.
Yep.
So I was wondering whether in these types of radicals, 1,2 spin center shift is a very common phenomenon. So did you observe any 1,2 spin center shift, or can you, is it possible to identify one under these reaction conditions?
Let me, maybe I need to go back to completely understand your question. Okay, let me go to this.
Yeah. Here, so after the cation, carbocation is getting trapped by 2-iodobenzoic acid,
Yes.
getting a secondary radical.
Yes.
So isn't it possible? So what happens usually in 1,2 spin center shift is this radical will get transferred to the primary carbonate, that is the carbon that contains the CO bond, and that CO bond will break, and you will get a carbocation on the carbon that is containing the radical, and that is now containing the radical, and you will get a radical that is on the carbon that is joined with oxygen. So you will get in essence you will get a radical cation, but in the reverse way. So did you observe the other product? So if you get the radical cation where the carbon joined with nitrogen contains the cation and the other carbon contains the radical, then you will get the nucleophile attacking on the carbon containing the nitrogen, and the electrophile getting trapped by the primary carbon radical. So did you observe any such kind of product?
No, we didn't any observe. I think the reagent selectivity was always perfect in this reaction, so we didn't observe the reverse product, and that was also something I was a little bit surprised, but it's confirmed also by a lot of work of David Nikiewicz. When you make this type of activation, you always observe this type of reactivity, or not the reversed one, where you have the reversed carbocation and the radical reactivity. So it's very clean into these directions.
Is there any specific reason for that, for the selectivity of the formation of radical cation?
It is I think a little bit surprising. I don't have really a very good explanation for that, because intuitively you would expect the odor to be a little bit more stable, but it could also be a kind of reactivity that is not the other one may be a little bit more stable, but it's much less reactive, because this one is certainly the more reactive.
Because if we would have got the other radical cation using, say, for example, electrochemical methods, then these two methods, the one that you showed with hypervalent iodine and the one with electrochemical methods, these two would have been orthogonal in nature, orthogonal with respect to reactivity. So
I try to think in the electrochemistry, but with electrochemistry, in theory, you could also access this type of intermediate, but I don't see why they would be orthogonal. That's something that would be different. Yeah.
For the sake of time, I'm trying to go in and just letting other people to see if they can ask a question as well. Are there any others who would like to contribute? And thank you so much for your question so far. There is no other right now, actually. But actually, we're also over time now. But it has been a true pleasure to host.
I think there is a last one now. I just see it.
Yvonne, yes, please. We can't hear you.
And now, can you hear me?
Yes, yes, yes.
Oh, good. So sorry, I cannot switch on my camera because I have no camera on this computer.
No worries.
But, so I enjoyed the talk very much. It's very interesting. I have one question on the decarboxylation reaction as well, the first part. I don't think you commented the choice of the base so much. I guess you need potassium or cesium to have more like a naked ion, an ion that would be easier to oxidize. But sometimes you use, I think, cesium benzoate, sometimes it's potassium carbonate. And I would just like to know how important it is. Does it make a big difference if you change the base?
Yes, let me go back just to the transformation. Okay. Yes. Yeah, that's an interesting question. First, there is a cesium effect. Definitely, there is a cesium effect, and it was discovered already by Macmillan that cesium give you a better yield in this type of transformation. But then, in their work, it was not so important to use benzoate or carbonate or things like that. But the benzoate was important specifically for the case of the cellular alkyne. The problem we have is that the cellular alkyne is a little bit sensitive to base, and the carbonate is a little bit more basic, and it kind of degraded the reagent and give you lower yield. But the cesium benzoate is fine to do the job, and is not degrading the reagent. So it was just that. So if you use aryl reagent, you can use cesium carbonate or cesium benzoate, and you get the same yield. But if you use the cellular reagent, then the benzoate is giving you the best yield. Yeah, that was the reason.
Okay, thank you. It's clear now.
Perfect. Maybe one last question, if anybody. Otherwise, we would say thank you for this presentation today. I don't think there are any more. Thank you so much, and looking forward for our next speak in 2 weeks from Technion. And then a little summer break for everybody. So thank you so much, Jérôme, and thank you for your presentation. We will publicize it on our homepage and on our YouTube channel. Thank you so much, and enjoy the rest of the day.
Thank you. Thank you for the invitation.